So one of the problems in AT is that it's currently an incurable disease, and there's currently no models available for in vivo testing of therapeutics, or even understanding exactly what's happening during the degenerative process. So it's important to consider new options like the zebrafish to see whether that can provide any clues that aren't currently available. So the funding that we've received from Action for AT is about £80,000, and what we want to do with this is to basically take on a PhD student who will be working for 3 years to get their doctorate, and their project will essentially be to genetically engineer some zebrafish so that they carry mutations that would give you AT if you were a patient, and then we will study what happens in those fish to see what happens to their cerebellum, what happens to other aspects of the AT phenotype. The other thing that the student will be doing is obtaining some zebrafish that have already been generated, which express this green fluorescent protein, which originally comes from jellyfish, and they express this green fluorescent protein in cerebellar neurons, and that allows you to put zebrafish embryos that are alive under the microscope and observe what's happening to the cerebellar neurons in real time. So potentially that could be a tool that we could use for screening to look for factors that enhance survival of cerebellar neurons in a disease model, and that could be applicable to any disorder affecting cerebellar neurons, so multiple forms of ataxia. So obviously we share a common ancestor with fish millions of years ago, and we'd look very little like fish today, but we share about 70% of our genes still with a fish. So in terms of a gene which is mutated in a particular disease, you've got a 70% chance that that gene exists in the fish, and if it exists in the fish, then it's highly likely to still do the same job in the fish as it does in humans. And then fish offer several unique properties as a model system, which is why we use them. So despite a lot of attempts to generate an animal model of AT, so far there's been limitations with every model that's been generated. So it seems that certain aspects of the disease can be modelled. So in terms of the genome instability aspect of AT, that seems to be faithfully modelled in mice. But the problem seems to be, the major feature of the clinical disease in man is ataxia and trouble with cerebellar function. And so far the mice that have been generated don't seem to show that at all. So if we're interested in developing therapies or gaining insight into actually what goes wrong in the cerebellum, then the mice currently can't really help us with that. We've been working with fish models of other cerebellar degenerative conditions, which don't cause AT, but they cause sort of related cerebellar degenerative phenotype. So we think that there's a chance that fish will be beneficial in the context of studying the genetic cause of AT, and that may allow us to develop an animal model which develops cerebellar degeneration, and then we can use that for understanding the mechanism and potentially identifying therapeutic approaches. Action for AT is fundraising to allow researchers to generate models and to investigate the mechanisms of disease in AT, and this is an area which is underfunded and really requires injections of cash to allow scientists to proceed with their research in this important condition.